All right, so guys, let's go quickly into this concept. This is us talking about the embryonic stem cells. We have the adult type of stem cells too. We have a lecture coming on it. All right, for this one now, we're basically talking about embryonic stem cells. Now, under the embryonic stem cells here, we'll be talking about the definition, characteristics, but we'll be focusing more on the impact that these stem cells have on biology and medicine. Okay, you can see this is what an embryonic stem cell. And what is a stem cell? If I could ask you, stem cell is basically any cell that's what is like an ancestor. It can break down to become any type of cell. Okay, so you can see that stem cells can become ectoderm, mesoderm, endoderm. The endoderm could become liver tissues, thyroid tissues. Mesoderm could become muscles and bones. Ectoderm, that's the outer ones, could become skin or nerves. Do you understand? So this is just a brief review of what a stem cell is. Okay. So embryos contain pluripotent what? Embryonic stem cell. When they say that something is pluripotent, that thing has the potential of becoming anything. Okay, so now it contains pluripotent what? Embryonic stem cells, which can give rise to all tissues of the human body. Okay, so embryonic stem cells can be maintained in culture as undifferentiated cells lines, or they can be induced to what? Differentiate into what? Many different lineages. Okay, so they can be maintained as what? Embryonic stem cells, no division, or they could be what? Induced to further divide into what? The needed type of cell. Okay, so I hope you are picking up. I hope you are following up, right? You see, these are what? Cultured undifferentiated stem cells, because they actually come from what? The embryo, that's a blastocyst. So these cells now, they have the potential to become liver cells, any type of cell. All right, this one is just for example. So these embryonic stem cells have been studied, have been used to study the specific signals and differentiation steps that is required for the development of many tissues. Okay, so now these cells are injected into blastocysts, which are then implanted into the uterus of a surrogate mother. All right, so now the genetically modified implanted blastocyst develop into full embryos as long as the gene defect does not cause embryonic death. Okay, so now you can see this. This is stem cells. They basically come from the blastocyst, and they have the potential to become anything at all. Okay, so now let's focus on what actually brought us here. Impact of embryonic stem cells on biology and medicine. Okay, so now techniques used for the genetic manipulation of embryonic stem cells have been greatly expanded in scope to produce gene deficiencies that are specific for a single tissue, and conditional gene deficiencies, that is, gene deficiencies that can be turned on and off in adult animals. Do you understand? So if you have a deficiency of a gene, you might actually derive that gene from a stem cell. Do you understand? That's what they mean. So knockout mice have been widely used model for experimental study of human disease and has provided essential information about gene function in vivo. Okay, so embryonic cells may, in the future, be used to repopulate damaged organs. It's still a study that has been going on. Do you understand? It's still a study that has been going on. So in the future, let me say you lost your hand, they could actually use stem cells, and if they can be able to induce the stem cells to produce the cells of the hand, your hand will regenerate. Do you understand? It's possible, although it's still under study. Okay, so in the future, it may be used to repopulate damaged organs such as what? Liver after hepatocyte necrosis and myocardium afterward infarction. All right. So now the generation of some specific cell types from the cultured embryonic stem cells has been what? Already been achieved. Okay. So now insulin producing pancreatic cells and nerve cells produced in these cultures have been implanted. That's basically talking about what? Experiments that they have done with these embryonic stem cells. So they have been implanted respectively into diabetic animals, in mice with neurologic defect. Okay, so now intense debate about the ethical issues associated with this type of therapy, known as therapeutic cloning, in man is ongoing. Okay, so they are still studying this, but at least you guys should have the knowledge of what an embryonic stem cell is and the impact of embryonic stem cells on what biology and medicine. So that's it and bye for now.